Good evening, fellows, and today we will start with the physiology of the renal system. I have covered the anatomy of kidney and ureter, urinary bladder, and urethra, and now I will discuss the functional aspect of these renal structures. Okay. First of all, start with the introduction. Introduction will consist of two parts, and after that, there will be four parts of GFR, the glomerular filtration rate. Okay, so let's get started. First is the table of content, the introduction, then function, mechanism of drug removal, and biotransformation reactions. Okay, what you understand with the renal physiology, it is the study on how the kidney maintains the internal conditions of the body, like osmotic conditions, homeostasis, and maintaining the pH of the blood, as well as the electrolyte imbalances. The functions of the kidney, there are four functions of the kidney: excretory, endocrine, metabolic, and metabolic functions, endocrine functions, and regulatory functions. Okay, first is the excretory functions. The As you all know that kidney excrete the metabolic waste products like urea, creatinine, toxin, drugs, and other harmful molecules which are dangerous to the body are eliminated by the renal system. Uh, how the kidney is functioning, to determine the functional aspect of the kidney, we don't use the urea level in the blood, because the urea level, if there is uremia, the urea in the blood, it is not only indicative of the renal failure, because it is also disturbed in case of sweating, in case of dehydration, polyuria, high protein intake. The urea level can be exceeded, but the gold test for the renal function is the serum creatinine, and you have to remember this test, because when the renal nephrologist suspect a kidney disease, they go for the serum creatinine, not for the urea. Okay, and the second thing I want to mention is how drugs are eliminated from the body. I know this is not relevant to the renal system, but most of the drugs are eliminated by the the kidneys. So I think it is in this case it becomes relevant to the topic. So, for the excretion or the elimination of the drug should be lipid soluble or water soluble. Think logically, because most of the cell membranes are composed of lipid bilayer. So, if the drug is more lipophilic, lipophilic means that it is loving the lipid, or it is more lipid soluble. So, it will cross the biological membrane and enter into the cell. So, it is not excreted, it enter into the cell, right? But when the drug is more hydrophilic, hydrophilic means that it is more water soluble, hydrophilic. So it likes to remain in the outside environment, not inside the cell, because it gets bounced back from the lipid bilayer. Okay, for the excretion mechanism, the drug must become water soluble, it must become hydrophilic. So, before discussing the functional aspects, let's discuss about the mechanism of drug removal. And this is known as the biotransformation reactions. Okay, just think logically that if we ingest a drug, it goes into the GIT, it goes into the portal circulation, and that then it goes into the general circulation. Okay, so the lipid soluble molecules, as I've discussed earlier, they can cross the biological membranes. It means the cell membranes or plasma membranes of the cell. For excretion, they must be hydrophilic, water soluble. So for drug removal, there are two systems: hepatobiliary, by which feces are formed, okay, and renal system, by which urine is formed. Okay, these two are excretory mechanism for drug, not only for the drug but also other toxic materials. For example, the food residues by the hepatobiliary, renal, and many other things like urea, bile, and other things are removed. Okay, but as we are discussing biotransformation, so the number one reaction and second, there are two reactions and they are separated on the basis of how the molecule became hydrophilic. So we are discussing first is the biotransformation one reaction. So the drug molecule is exposing a charge group. You know that ions, which the ions are more water soluble. For example, if there is an ion, it can cross the plasma membrane easily as compared to polar substances, because the polar substances cannot cross the cell membrane. Sorry, charged molecule can also not cross the biological membrane. So because it became water soluble, I'm discussing here, they became water soluble when exposing a charge group or modifying the molecule to express a charge group. So the lipophilic drugs are converted into water soluble drugs. Okay, the second reaction is that the lipid soluble molecule, for example, is a drug. It is combined with highly charged molecule secreted by the hepatocytes, and it can be glucuronic acid, the one of the molecule which is associated with the glucose, and it became a polar complex because it has combined with a charged molecule. So what's the difference? In biotransformation, a one reaction, the molecule expose a charge group, so it becomes hydrophilic. In biotransformation second reaction, it is combined with a charged molecule, for example glucuronic acid, and then it becomes easy to excrete. As I've told that polar molecules which are water soluble, they are not entering into the cell, so they must go to the excretory systems, the duodenum and the nephrons. They go to the duodenum by hepatobiliary system, they goes to the nephron by the circulatory system and excreted as feces in the hepatobiliary and urine in the circulatory or nephritic system, nephrons. Okay, let's discuss by the diagram. We ingest something, maybe a drug molecule or food molecule or something like anything. Our food is a complex of many things. So it goes by the portal circulation. First, it goes by the portal circulation into the liver. These are the hepatocytes. After a modification in the liver, water and solutes, they either choose one of the two pathways. They can go into the biliary system or they can go into the general circulation. From the biliary system, they goes into the duodenum, to the large intestine and to the feces. In the general circulation, it is ultimately going to the nephrons, and by the tubular system they are excreted as urine. Okay, I hope you understand the biotransformation reactions. The first is expressing the charged molecule, and second is combining with a charged molecule to become more polar, to be excreted. This was the excretory function of the kidney. Let's discuss the regulatory function. Regulation means that the kidney is maintaining the fluid, the electrolyte balance, and the pH of the body, means blood. It must maintain in them within a narrow range. So, electrolytes. First is the sodium. I will discuss the main of them. There are there are many of them, but the relevant to the laboratory test and for you is only these. I think these would be pretty enough. First is the sodium. Sodium should be maintained in 135 to 145 millimole per liter. Potassium 3.5 to 5.5. Chloride 100 millimole per liter. Bicarbonates 22 to 28 millimole per liter, and your favorite, the calcium, 2.5 millimole per liter. And millimole per liter is also can be equivalent to the milli equivalent per liter. Okay, this is also unit used in some countries, milli equivalent per liter. So um, the potassium should be maintained within a very minor and narrow range, because if there is hypokalemia, which means less than 2.5, so the 3.5 and 5.5 is the range. But when it exceeds 6.5 or it is less than 2.5, it becomes hypokalemic and hyperkalemic respectively, and they can induce dangerous arrhythmias. Arrhythmias means that irregular beating of the heart, irregular cardiac rhythms. So they can induce cardiac arrhythmias, and it can be very fatal to the body. Okay, sodium, potassium, calcium, bicarbonate, and the chloride. So the the third function is the endocrine function. Endocrine function is to secrete the hormones into the blood, which then act on the target cells. There are three endocrine functions you must remember relevant to the kidney. First is the erythropoietin, the second is renin, and third is the prostaglandins. Erythropoietin. Erythropoietin is a hormone that triggers the RBC's production. As the name suggests, it's erythropoietin. It is produced by the kidney, by specifically by which part of the kidney? By the endothelial cells of peritubular capillaries. What are peritubular capillaries? As you know, that this is the glomerulus. Okay, this is the efferent arteriole. This is the efferent arteriole. Okay, this is efferent and this is efferent arteriole, and these are the proximal tubule, convoluted tubule, then loop of Henle, then distal convoluted tubule, and the collecting duct. This diagram is not anatomically correct, but I'm just putting the concept in your minds. The efferent arteriole then wrap around the peritubular capillaries, the proximal convoluted tubules, and the distal convoluted tubules, and these are known as peritubular because they are surrounding the tubules. Erythropoietin is produced by the endothelial cell of these peritubular capillaries. Okay, it is a concept that on how demand the erythropoietin is produced from these cells. First of all, oxygen. Oxygen is a major triggering factor for the erythropoietin. Let's see how it can trigger. Oxygen from the nephrons, okay, it activates the oxygen sensing proteins in the endothelial cells of peritubular capillaries. These protein sensors, they goes to the nucleus of endothelial cells of peritubular capillaries and block the erythropoietin producing gene. Why it blocks them? Because the oxygen is already present in the body, so oxygen levels are high. We don't need RBCs. We don't need enough oxygen. We are not hypoxemic. We have enough oxygen, so we don't need erythropoietin. So, according to the feedback, the oxygen blocks the erythropoietin. But on the contrast, if the person is anemic, it has less RBCs, it has less oxygen, it has less hemoglobin, so the body become hypoxemic. And in this condition, the oxygen, the inhibitory sensors will not work due to less oxygen. When the inhibitory sensors are not working, so erythropoietin producing genes are released. Let's discuss in this diagram. If the person is anemic, the RBCs levels are going down, oxygen levels are going down. These are the inhibitors. The inhibitors which are inhibiting the gene. But when the oxygen is less, inhibitors are not working because they also need oxygen for their proper functioning. When inhibitors are not functioning, the gene goes out of the nucleus by the process transcription. After translation, it converted into erythropoietin hormone, via the blood it goes to the bone marrow. In the bone marrow, it acts on the erythroid series, which are the precursors for RBC production, and ultimately RBCs are produced. And we need them because they are carrying oxygen. And we were anemic, we were hypoxemic, we have less RBCs, we have less oxygen. So ultimately, via the positive feedback, RBCs are producing more. So it goes to the bone marrow and acting on the erythroid series, and RBCs are being produced. Also, genetically engineered erythropoietin is also available, and they are injected into the anemic patients with renal failure. Okay. The second hormone which is produced is the renin. There is a separate video I will make on the RAS system, and just for now, remember that renin is produced by juxtaglomerular apparatus. In the upcoming video, I will discuss where is the location of this apparatus, what is RAS, which is renin angiotensin aldosterone system, and how it works. Okay. The third is the okay, sorry. My cat was disturbing, so let's continue. The third thing is the prostaglandins. Prostaglandins are the third hormone or proteins which are produced by the kidney. What is the function of prostaglandins? These are vasodilators. Vasodilators means that they dilate the blood vessels and they regulate the intrarenal blood flow. They maintain the blood flow to the kidney. Okay, and these are very important physiologically. NSAIDs, what are NSAIDs? NSAIDs are non-steroidal anti-inflammatory drugs, are contraindicated in renal failure. Why they are contraindicated? Because they can reduce the blood flow to the kidney by inhibiting prostaglandins. So they can induce ischemia. They induces the reduced blood flow, so they can induce ischemia. So they are contraindicated in the patients who have kidney disease or who have renal failure. Okay, the fourth function is the metabolic function. Let's zoom it a little bit. Okay, it's the last. Finally, metabolic function is there are two metabolic functions which are provided by the kidney. A lot of them, basically, for example, renin, it maintains the BP. It is also considered a metabolic function, but the two important one are: first is the conversion of inactive vitamin D into active form. You all know that our skin, on exposure to the sunlight, produces cholecalciferol, but cholecalciferol is not enough. It must undergo several conversions before it converted into active form. The first conversion, or in more technical term, hydroxylation, occurs in the liver. Hydroxylation means, what means hydroxylation? It means addition of hydroxyl group, simple, and it is added on the 25th position of cholecalciferol, so it becomes 25 hydroxy cholecalciferol, but it still is inactive. Second hydroxylation, when it comes the kidney, in the proximal convoluted tubule cells of the kidney, they have an enzyme known as alpha 1 hydroxylase enzyme, and this enzyme adds another hydroxyl group or hydrogen group into the first position, so it becomes 125 dihydroxycholecalciferol, and it is the active form of vitamin D. And good news, the vitamin D can absorb through GIT mucosa, and it can have a lot of functions to strengthen the bone, calcium absorption, and all of that. The other thing worth mentioning is the parathyroid hormones act on these cells to activate alpha 1 hydroxylase. When alpha 1 hydroxylase become activated, it adds the hydroxyl group to the 25 hydroxycalciferol, and it becomes active form of vitamin D. So in this condition, parathyroid hormone is influencing for the production of vitamin D, active form of vitamin D. Okay, I think you understand. First is the cholecalciferol. In the liver, first hydroxylation occurs. It becomes 25 hydroxycholecalciferol. In the PCT, an enzyme converts 25 to 125 dihydroxycholecalciferol, and parathyroid hormone have positive impact on these cells. Second metabolic function is the glucose metabolism. It is very interesting to know that in fasting conditions, not normal, but during prolonged fasting, we are deprived of glucose, and we have consumed all of the glucose present in our body. So we must synthesize new glucose, which is known as gluconeo, means new, genesis means producing. But the part worth mentioning is that gluconeogenesis uses non-carbohydrate sources for the production of glucose. For example, amino acid, fatty acids, like these are non-carbohydrate sources. Understand? Okay. So, um, I hope this is enough for today's lecture. And just revise a little bit. I have skipped the one more function, and I will discuss it in the next video, which is also introduction based. So this all works with the physiology of the kidney. Physiology does not only means the renal functioning, the glomerular filtration rate and all of that, but it also includes these functions which are normally excluded in many textbooks, but it is worth mentioning. Okay, so you can take screenshot for your making own notes, or as you wish you can do. So let's meet next time in the next video. Till then, take care and goodbye.